Okay, so cell walls. Are cell walls unique to plants? Are there other organisms that have cell walls? Okay, so basically it's everything except for animals have cell walls. So why cell walls? What roles do cell walls play, not just for plants, but for any organism? Hemi? Protection in what sense? So would it prevent cadmium ions from getting into the cell? Okay, so I won't be a little bit more specific by what you mean. Change your mind.
Like it keeps maintains a certain volume. So.
Okay. If we want to be more general, we might say shape, right? What else? For a plant cell, would the plasma membrane determine the volume of the cell? So if the cell was under isotonic conditions, same osmolarity in the cytoplasm as outside the cell, yes, then the plasma membrane could determine it. But under most circumstances, plants are in medium that has lower ionic strength than the cytoplasm. If there's just the plasma membrane there, the cells would rupture, right? So doesn't that tell us something about what the cell wall does? What is that? Say that again? Yeah, so basically it constrains the turgor pressure. Or resists the turgor pressure. And we also should think about, in the context of many plant cells, the turgor pressure is what keeps the cell having a shape, right? If the turgor pressure drops to 0, or we have plasmolysis, if you have a flower that's where the turgor pressure drops to 0, it wilts, right? It loses its structural integrity, right? So there's some mechanical components to this as well. So cell walls that have secondary walls that have things like lignin and stuff like that in them are mechanically strong. Even when the cells are dead, they still maintain their shape. So things like xylem, right? What else? So you talked about protection. Protection against what? What sort of things do cell walls protect? Pathogens, herbivores even, maybe? Yep. What other role in protection did we talk about for cell walls?
It's not exactly cell wall, but the wax layer prevents water.
Yeah, so the waxy surface of the cuticle on epidermal cells prevents water loss. Back to the protection against the pathogens. What other thing did we say about, besides sort of a mechanical, structural protection against pathogens, what other role does the wall play in pathogens? If you're a pathogenic bacteria or fungi and you want to get into the cytoplasm, what do you need to do to get into the cytoplasm of the cell? Before you even get to the plasma membrane, what do you need to do? Get through the cell wall, right? So what does the bacteria or the fungus have to do to the cell wall? Degrade it. And what do those degradation products do? Yeah, they're signals, they're elicitors, right? So we should also keep in mind this elicitor function, that breakdown of the pathogens of the plant cell wall by pathogens release products that are signaling molecules that turn on defense pathways. What else? We're missing one other important thing in terms of, let's say, transport. What role does the plant cell wall play in transport? Say that again? Yeah, apoplast. So the fact that the cell wall is very porous and very hydrophilic means that it plays an important role in apoplastic transport. Okay, the one other thing that we really should be thinking about goes back to the top of this list. Not only does the cell wall determine the final volume and final shape of the cell, but remember, or if you don't remember, we'll talk about it in lecture Thursday. Once you get past the embryonic stage of a plant, once you get past the seed, all the new cells are derived from the meristems. What is the size and shape of a meristematic cell? What's the size of a meristematic cell? They're really small. And are all the other cells in a plant really small? Xylem cells, phloem cells, they're pretty big. Epidermal cells, they're flat and shaped like jigsaw puzzle. What determines how that meristematic cell expands to get to the final shape of the cell? The cell wall. Right? The cell wall is what determines how the cell enlarges, how it changes its shape. So it's not just determining the final shape, but it's determining the path that it follows from the little cubicle cells that are in the meristem to whatever the final shape of the cells are from the mature cells. Are all cell walls in the same plant the same? No, of course not. So if we think about xylem, for example, xylem has heavy walls that resist the cell collapsing under the negative pressure that's present in there. Phloem resists the positive pressure in there. Okay, so those have very heavy cell walls that give rise to the woody characteristics of plants. They're mechanically strong. We talked about the epidermis and the presence of the cuticle, right? That's a specialized component of the cell wall that's only found in the epidermis. How about the endodermis in the roots? It's got that suberin layer that makes up the Casparian strip. So cell walls vary from different types of cell within the plant depending upon the properties of the cell. Even within a single cell, the cell wall can be different. On epidermal cells, the cell wall is much thicker on the outside of the epidermal cell than it is on the inside. How about guard cells in the stomates? Remember when the pressure changes in the guard cell buckles and opens up the pore in the stomate? That's all due to the properties, the asymmetric properties of the cell wall. We talked about companion cells, that different types of companion cells have different types of plasmodesmatal connections. So in apoplastic phloem loaders, there's no plasmodesmata between the bundle sheath and the companion cells. And in symplastic phloem loaders, there are plasmodesmata there. Okay? So these wall characteristics can vary from cell type to cell type depending upon the function of the cell. Right? So we should view the cell wall not as being this static thing that constrains mature cells, but rather as being a very dynamic thing in the process of cell development. We start off with all the cells being these little meristematic cells, and we end up with all these different types of cells, many of which have different cell wall characteristics, most of which have different shapes and sizes, all determined by characteristics of the cell wall. Okay. We should also recognize that the cell wall, studying the cell wall, is a really hot area of research in plant biology these days for two reasons. One, cellulose, biofuels, right? People want to find mechanisms of converting cellulose into glucose that can then be fermented to make alcohol. If you think about the current way we make most of our biofuels, ethanol-based biofuels, it's from edible stuff, starch basically, from corn. And it makes a whole lot more sense to make biofuels from inedible stuff, like cellulose. So that's a very hot area of research. And obviously we also talked about the roles of the cell walls in pathogen protection. So varying cell wall properties are also important in trying to engineer differences in pathogen protection. Finally, if you're an ecologist, cell walls are very important in ecology. Why? What is it about cell walls that make them so important in ecology? Hemi?
The book says it's the biggest organic food.
Oh, yeah, so you remembered what the book says. Yeah, so that's correct. It's the largest pool of organic carbon in the biosphere. Why is that? Why are cell walls the largest pool of organic carbon in the biosphere? Well, clearly there must be more cell walls in detrital material in the soil than there are cell walls in living plants. Otherwise, the cell wall stuff wouldn't be the largest pool. That's right. They don't degrade easily, right? The fact that they don't degrade easily means after the plant dies, the cell wall material is relatively refractory. It's not broken down easily by organisms in the soil. So it tends to accumulate there. So the largest component of organic carbon in the biosphere is the non-living remnants of cell walls in the soil. Okay. Much more carbon there than there is in the cell walls of living plants. I mean, orders of magnitude more carbon there. Okay, so let's think about cell walls in terms of their general properties. If you look at an introductory to biology textbook, pictures of cell walls tend to emphasize three different layers in the cell wall. There's the middle lamella, this is laid down first. Let's think about this in terms of cytokinesis. A cell dividing to form two daughter cells. When those two daughter cells form, you must form a new cell wall in between the cells. The first layer of the cell wall that's laid down, the thing that's in the middle of the cell wall, is called the middle lamella. The middle lamella is basically glue. It's the thing that holds the cells together. In terms of composition, it's mostly pectins. What are pectins used for? In humans, what do we use pectins for? Yeah, jams and jellies. It's a thickener that we put in jams and jellies. But in plants, it's the same material. It's just the thing that holds the cells together. When a fruit ripens, think about this being the cell wall between two cells in the fruit of an apple. When a fruit ripens, what happens is that pectin breaks down. Now, if the fruit is nice and crisp, you bite into it. This is good and solid. When you bite into it, you rupture the cells. You taste the nice sweet part inside the cell. When the fruit gets ripe, when it gets mushy, the reason it's mushy is you bite into it, this pectin layer is broken down. The cells just slide past each other. Your teeth aren't close enough together to crunch individual cells. So the reason an apple tastes mealy when it's overripe is because the pectin is broken down in the middle lamella. Okay, the next layer as we think about the sequential growth of the layers, is the primary wall. The primary wall contains cellulose. And the easiest way to think about the primary and secondary walls is it's like fiberglass. Fiberglass has got glass fibers that give tensile strength that are embedded in a matrix. Well, cellulose is the thing that gives the cell wall tensile strength, and the matrix kind of holds it all together. We'll talk in more detail about these components in just a minute. But the primary wall is the part of the cell wall that plays the biggest role in development. The primary wall is what dictates how the cell expands and changes shape from that little meristematic cell to whatever the shape of the mature cell is going to be. So that tells us that the primary wall has to be flexible. Not flexible in terms of bending, but flexible in terms of being able to allow the cell to take on a lot of different shapes and sizes. And as I said, we'll talk more detail about that. So this is present during cell growth, or laid down during cell growth. Once cell growth is ceased, the secondary wall is produced. It also contains cellulose, but it also typically contains compounds like lignins. And basically, once the secondary wall is laid down, the cell no longer has the ability to expand. Okay, so this is after growth ceases. So in a developmental sense, the middle lamella is laid down immediately after the cells divide. And it's the glue that holds those cells together. The primary wall is laid down pretty quickly after this, but the primary wall can change its characteristics to allow the cell to grow, to get to its final mature shape. And once it gets to that shape, then the secondary wall is laid down. The secondary wall might be very thin in a number of cells and can be very thick in other cells. So this is the most variable component of the cell walls. The middle lamella and primary wall are characteristics of all cells and are generally pretty similar. The secondary wall differs greatly depending upon the characteristics of the cell. I don't know the answer to that, but my guess is at least most of them do. There probably are some examples of cells that don't make, well, okay, so give me an example of a cell that doesn't make secondary cell walls. Meristematic cells. That would be the first thing that you'd think of, right? But they certainly have primary cell walls. What would happen to a meristematic cell if it didn't have a primary cell wall? Yeah, it would burst, right? There would be no way to constrain the turgor pressure. Right? Okay, so let's think about cell walls from the context of cell division. So here we got a cell in anaphase where the chromosomes are separating from each other. In telophase, we have the daughter nuclei are starting to form. And this is a picture from the last version of the textbook. Figure 1.25 in the new version of the textbook still hasn't corrected a major error that they have. One of the things that dictates the formation of the cell wall between the two new daughter cells is that we know that these microtubules that play a role in separating the chromosomes during mitosis, as soon as those chromosomes are finished separating, the majority of those microtubules reorient in a direction that's perpendicular to the way the chromosomes just move. They reorient in the region where the cell wall is going to form. And this aggregation of microtubules is called a phragmoplast. And basically these microtubules form the skeleton upon which the new plasma membrane that separates these two cells and the middle lamella will be formed. And it's relatively straightforward to think about how this process happens. So basically, between the two daughter cells, we have this network of microtubules, the phragmoplast. And what these microtubules do is they attract vesicles derived from the Golgi. So vesicles derived from the Golgi tend to aggregate along these microtubules. And these vesicles, here are two vesicles, what happens is they fuse, forming larger and larger vesicles. And eventually, this larger vesicle fuses with the plasma membrane on either side of the cell. When that happens, two things have been accomplished. The membranes of those Golgi-derived vesicles provide the membrane that is now the plasma membrane that separates the two cells. The contents of these vesicles is the middle lamella. As these vesicles fuse, that contents is left in between those vesicles where they fused. Okay, so the fusion of those Golgi-derived vesicles does the two things that need to happen during cytokinesis in plant cells. It provides the membrane that gives rise to the two new plasma membranes that separate the daughter cells. The fusion of those vesicles provides that membrane. Those vesicles eventually fuse with the plasma membrane of the parent cell, giving rise to the two daughter cells. And all the stuff that was inside these vesicles are the pectins that make up the middle lamella. Yes?
So they don't form. So is the middle lamella a membrane?
No, the middle lamella is carbohydrates. Pectins are a bunch of carbohydrates that are very sticky.
So it's like, I'm just trying to figure out how this happens. So,
Okay, so here's some vesicles that are derived from the Golgi that are coming up here, and they fuse together, right? So as they fuse together, they form larger and larger vesicles. Once this vesicle fuses with the plasma membrane, then you've got plasma membrane continuous to the vesicle all the way over, fuses on the other side. You got one plasma membrane here and the other plasma membrane there, okay? And then all the stuff that was enclosed in those vesicles becomes the middle lamella, right? So two things are accomplished simultaneously by these Golgi-derived vesicles and their fusion along this phragmoplast, right? And that should make it clear to you why the middle lamella is in the center of the wall. It's laid down first. The primary wall is laid down by each cell next. The secondary wall is laid down by each cell last, right? So the most recently synthesized part of the wall will be the part that's closest to the plasma membrane of the cell that made it. Okay? We all right with this? Okay, so let's think more generally about the overall synthesis of the cell wall, because basically we said the main part of the cell wall, once we get past the middle lamella, consists of two different types of materials. It consists of cellulose, the analogy to the glass fibers in fiberglass. They're the things that give the wall tensile strength. And they include matrix materials, the resin in which these microfibrils are embedded. And these two components of the cell wall are synthesized by different mechanisms. Okay? If you think about cellulose, it's this long polymer of glucose. And it's, in general, in the cell wall, it's not a single polymer, but it's aggregates of polymers that make these bundles. These things that show up here in gray in here are the cellulose microfibrils. We'll see in just a few minutes that these microfibrils are bundles of cellulose molecules. The way they're synthesized is that there is a complex in the plasma membrane that is synthesizing them in the plasma membrane and extruding the cellulose microfibrils outside the cell. So the cellulose does not exist in the cytoplasm. It's made in complexes that are embedded in the plasma membrane, and as they're made, they're extruded outside the cell. So the microfibrils have one mechanism by which they're produced, by these cellulose synthase complexes that are present in the plasma membrane. All the rest of the matrix materials are delivered to the cell wall by the same mechanism that the middle lamella is sent to the cell wall during cell division. That is, there are vesicles derived from the Golgi that carry the components of the matrix materials. When those vesicles fuse with the plasma membrane, those matrix materials are released out into the cell wall. And so one of the things that you might think about the characteristics of the cell wall is all the basic construction is done inside the cytoplasm. What holds the wall together? Are there covalent bonds that are formed out here when the cell wall is being formed? And the answer to that is, in general, no. Most of the cell wall is held together by hydrogen bonding, non-covalent interactions. There are some covalent ones, but they're the minority. Most of this is held together by non-covalent bonds. And you want to think about how this non-covalent interaction gives rise to that first thing we put on the list up here. That is, the ability of the primary wall to change its size and shape to dictate how the cell expands. Right? So that non-covalent characteristic of at least of the primary cell wall, permits the cell to expand. And we'll look at this in more detail in just a second. Okay. So, in terms of what the cell wall is made out of, well, the cellulose is just polymers of glucose. But all the matrix materials, the Hemi Celluloses and the Pectins, are made up of a wide variety of different types of monosaccharides that have been converted into acids, so sugar acids like glucuronic and galacturonic acid. And also, there's a number of deoxy sugars, not deoxyribose like in DNA, but other types of deoxy sugars. There are literally thousands of different types of sugars that show up in plant cell walls. Why the diversity? Why so many different types of sugars?
Maybe, could it be like a defense mechanism?
In what sense?
If, like, say an insect has like enzymes to break certain kinds of bonds, but maybe not others, so
Yep.
the diversity.
Okay, so the diversity helps limit the ability of pathogens to break down the cell wall. Yes, that's certainly a big component of it. Another component of cell walls you should think of in pretty much the same context as secondary products. Remember we said plants make lots of different types of secondary products, and one perspective is that they do that just in hopes of it being something that's going to be good for the plant. The evolution of the different pathways that form secondary products are not being guided toward a specific end product. They're random. The evolution of the metabolism that produces all these types of sugars that function in plant cell walls follow the same sort of thing. So although some plants will contain, some sugars will be found in all plants. So glucose is found in every plant cell wall. But there are other types of sugars that are unique to a certain lineage of plants in the same way some secondary products are unique to them. Okay, so that the characteristics of cell walls, you could use the characteristics of cell walls as a phylogenetic key to determine this plant is in this family and this plant is in this family. Because some of the monosaccharides that are present in the cell wall follow these phylogenetic patterns in the same way that secondary products do. Okay. So if we think about the components of the cell wall, here's a nice picture of the cell wall after most of the matrix material has been removed. So what you're basically looking there is a mat of the cellulose microfibrils. Okay? And one of the things that we'll talk about in a few minutes is what determines how these microfibrils are laid down, the pattern that they're laid down. It looks pretty random here, right? But we'll see that that's not always the case. But in terms of the characteristics of the microfibrils, remember we said that the cellulose microfibrils are the things that give the cell wall its tensile strength, its ability to withstand the turgor pressure. It doesn't stretch well. Those microfibrils can bend pretty easily, but they don't stretch very well. And we need to think about how those characteristics arise as a result of the molecular properties of cellulose. So cellulose is just a polymer of glucose. And a single cellulose molecule by itself is not that strong. But when you bring many cellulose molecules next to each other, the thing that can happen is, remember, all those glucoses have lots of hydroxyl groups sticking off them, right? Hydroxyl groups easily form hydrogen bonds. So what you can end up with is a very large network of hydrogen bonds among adjacent cellulose molecules. So within this cellulose microfibril, the cellulose molecules themselves form regions that are highly hydrogen bonded. Interestingly, when they first started looking at the characteristics of cell walls, it was about 50 years ago now, what they found was some regions of the cellulose are highly ordered. They scatter X-rays like the cellulose molecules are almost crystalline. So they describe some regions of the cellulose microfibrils as being crystalline domains, that is, highly organized in terms of a regular structure of hydrogen bonding. Other regions are less organized, which basically means there's fewer regular hydrogen bonds, but there's still a lot of hydrogen bonds here. So what gives the cellulose microfibril its strength is not the covalent linkage along a single chain of glucose molecules, but the hydrogen bonding among 20, 40 to several hundred cellulose molecules that make up this microfibril. Okay, so this is something that can bend but cannot stretch very well. And we'll see that becomes important in terms of the characteristics of the cell wall. Okay? So if we think about how the cell wall is put together, here's a sort of a cartoon figure from your textbook. Here's, in the green, are the cellulose microfibrils, so these bundles of cellulose molecules. And what these darker green stringy things represent are the Hemi Celluloses. They're also long polymers, although they're very often branched polymers, of lots of different types of sugars. But the role that they play is that they can form covalent bonds with one cellulose microfibril and then branch over and form covalent bonds with another one. Sorry, hydrogen bonds with another one. So the hemicelluloses behave like the cellulose, except they form linkages between adjacent microfibrils. And these linkages are held together by non-covalent hydrogen bonding interactions. Remember, they're all basically sugars, so they have lots of hydroxyl groups coming out here. These black squiggly lines here are meant to represent pectins, although the middle lamella is almost all pectin. There's pectins in the matrix of primary and secondary walls. And pectins are acidic. They're made of glucuronic and galacturonic acids. Acids under physiological pHs tend to have positive charges on them. And these little dots in here we'll see represent calcium ions that form ionic interactions that help hold the cell walls together. That is, pectins that interact with two different cellulose microfibrils are themselves held together by ionic bridges from the negative to the positive to the negative charges associated with the calcium and the pectins. And then these red things here represent structural proteins. Structural proteins are a very different component of the cell wall. Structural proteins do form covalent linkages with the various components of the wall. With the cellulose microfibrils, with the hemicelluloses, not so much with the pectins. So there are some covalent linkages there, but they are largely restricted to the structural proteins. These structural proteins tend to be highly glycosylated. They have lots of sugars attached to them, and those sugars form non-covalent interactions with all the other stuff there as well. So what you should take away from this picture is that most of the interactions that give rise to the overall structure of the cell wall are non-covalent interactions. Covalent bonds hold each of these individual polymers together, but the polymers are held together by largely by non-covalent interactions. Okay, so let's think about how these cellulose microfibrils are made. Right? We said that they're big things that function outside the cell wall. They're not synthesized inside the cell and then exported out. They're made at the level of the plasma membrane, and as they're made, the product ends up outside the cell rather than inside the cell. And the first evidence of how these cellulose microfibrils were made came from looking at electron micrographs of plasma membranes of cells that were rapidly growing. If they're rapidly growing, they must be producing a lot of primary cell wall to keep a cell wall around the cell that's getting bigger. And what they noticed was these little sort of rosette patterns. So here's a blow-up of them, although I think you can actually see it probably a bit better here. And it became clear that these rosette patterns were at least well correlated at the time these studies were done with the presence of machinery that makes a lot of cellulose in primary cell walls. And it turns out these little rosettes are basically aggregates of a protein complex that's involved in cellulose synthesis. So the rosette itself is made up of a bunch of rosette subunits, which is made up of an individual enzyme, which is the cellulose synthase enzyme. Cellulose, remember, is just a polymer of glucose molecules. And the fact that these things form this rosette means that all of the cellulose microfibrils that are formed by each one of these individual cellulose synthase enzymes end up together forming a single bundle. So depending upon the type of plant you're talking about, there could be 20 or 40 cellulose synthase complexes in a rosette, or there can be several hundred, depending upon the size of the microfibrils that are being produced by these complexes. Okay? Now, one of the interesting questions that you can ask about this cellulose synthase mechanism is, where's all the carbon coming from? One of the things that we learned earlier is that most of the carbon that is being exported from the leaves to the rest of the plant is exported in the form of sucrose or one of the larger trisaccharides or tetrasaccharides that are transported in some of the symplastic phloem loaders. Sucrose is actually the substrate that is used to build the cellulose molecules. Remember, sucrose is a disaccharide of glucose and fructose. So the first thing that has to happen if we're going to use sucrose as the source for the glucose molecules to make cellulose is you have to break the sucrose down. Normally, the enzyme that breaks down sucrose associated with metabolism, we said, is an enzyme called invertase. It takes sucrose and just hydrolyzes it, adds water in and makes glucose and fructose. That is not what happens in the cellulose synthase complex. In the cellulose synthase, what happens is there's an enzyme we talked about, sucrose synthase, that takes UDP glucose plus fructose and makes sucrose. In the cellulose synthase complex, that same enzyme is running backwards. It's taking sucrose plus UDP and making fructose plus UDP glucose, and then that UDP glucose, which is already activated with the UDP, is ready to make the glucose polymers in cellulose. So remember, when sucrose was being synthesized or when starch was being synthesized, that the monosaccharides had to be activated first before you could form the polymer bond. We formed either UDP glucose or ADP glucose. The same is going on here. The activated form is what of the sugar is what's used to make the polymer. So to make that activated form, sucrose synthase is basically running backwards, breaking down sucrose and making UDP glucose and fructose, and the fructose just goes back into the cytoplasm as part of the metabolism. So one thing you'd expect in cells that are rapidly synthesizing cell wall materials is that fructose levels in the cytoplasm might be higher because of all the fructose that's being recycled associated with cell wall synthesis. Okay. Questions on this before we go on? Remember, this complex is in the plasma membrane. Cellulose never exists inside the cell. As the cellulose polymers are being produced, they're being produced on the external surface of the cell, not the internal surface. So you never see cellulose in the cytoplasm. Okay. So one of the questions that we need to ask, and a number of you ask questions about this in your reading assignments, is the cell wall, the microfibrils in the cell wall we'll see play an important role in how the cells grow and expand. The question is, what determines how the microfibrils are laid down? And the answer to that is we don't really know that well. But there's a lot of circumstantial evidence that here is the cell wall with cellulose microfibrils, the newest ones. These are closest to the plasma membrane. Here's the edge of the plasma membrane where it's been torn away in this micrograph. And then here is a cortical microtubule, part of the cytoskeleton, that is just inside the plasma membrane. And you'll notice that there is a correlation between the directionality of the microtubule inside the cell and the direction of the most recently laid down cellulose microfibrils in the cell wall. This was the first evidence that the microtubules that lie just underneath the plasma membrane on the cytoplasmic side are acting as guides for these rosettes to move along, that are determining how the cellulose microfibrils are laid down in the cell wall. There's been other experiments that have given more circumstantial evidence. There are drugs like colchicine that causes the depolymerization of microtubules. If you treat a cell with colchicine, the newly formed microfibrils of the cellulose in the cell wall are all randomly oriented. They lose any specificity. So it again suggests that the orientation of the microtubules is what's determining the orientation of the newly formed cellulose microfibrils. But the mechanistic link between those is not understood. So it's still circumstantial evidence. But it should be clear, if it isn't clear to you now, it'll be clear to you by the end of the lecture, that something must dictate the orientation of the cellulose microfibrils, because they play such an important role in how cells enlarge, how cells change shape. So something has to be controlling it. It appears to be the microtubules that lie just underneath the plasma membrane in the cytoplasm. Okay. So let's switch gears for just a second to talk about how the matrix materials are made. We've already given the main clues to this. The matrix materials are made largely in the Golgi apparatus. So in the lumen of the Golgi, there's lots of machinery for synthesizing carbohydrate polymers. Those carbohydrate polymers are then bundled up in the interior of vesicles that bud off from the Golgi, move to the plasma membrane, fuse with the plasma membrane, and when they fuse, the contents of those vesicles is released into the cell wall. So all the hemicelluloses and pectins and things like that are delivered to the cell wall by this mechanism. Once they're in the cell wall, the assembly of this structure, to a large extent, is automatic. It's non-enzymatic because most of the interactions that hold this together are non-covalent interactions. Right? So the assembly of the cell wall is not something that requires a lot of enzymes. There are a few. We'll see some examples of it in just a minute. But for the most part, the materials are just delivered and they sort of diffuse around until they form the right number of hydrogen bonds and then they're stuck in place. Okay, so there's not a lot of machinery that's involved in the assembly of the cell wall. It's largely automatic, based on the properties of the molecules that are being produced there. Lots of hydroxyl groups, lots of capacity for hydrogen bonding. Okay, just some examples, pretty pictures from your textbook. This is a Hemi Cellulose. That's actually 4 different molecules that are present here. And one of the things that this shows that I want to remind you of, this is a borate ion. Remember when we talked about essential nutrients for plants? We said that boron is an essential nutrient. This is one of the reasons why boron is important. It's required for the assembly of a large variety of hemicelluloses that function in plant cell walls. If you take away boron, you take away the ability to produce a good deal of the matrix material that holds the cell walls together. So boron deficient plants can't make cell walls as effectively as they can in the presence of boron. So all the different colors represent different types of monosaccharides that are involved in making this polymer. And these can be 10s or 20s of monomers, or they can be tens of thousands of monomers, depending upon the type of hemicellulose we're talking about. Pectins are a little simpler. They tend to be chains of their polygalacturonic acid. So you'll notice that all these sugars have an acid group associated with them. Remember that acids at physiological pH tend to have a negative charge. So one of the things that holds, that allows the middle lamella to hold the cells together are these ionic interactions where the negative charges of the acid groups of the pectins are electrically attracted to divalent cations, and particularly calcium. So if you have plants that are growing in calcium deficient soils, not only does the lack of calcium cause problems for example, signal transduction pathways in the cytoplasm, they also have significant problems in terms of the cell walls because these calcium ions play an important role in holding the pectins together. And remember, the pectin is the thing that holds basically the glue that holds 2 adjacent cells together. Okay, what's most of the cell wall made out of? Water, yeah. Forget that part about it. Remember, it's this, basically we describe the cell wall as a sponge. All the hollow areas of the sponge are filled with water. Typically, cell walls are 80 to 90% water, right? That's certainly an important component of cells in terms of their interaction with their environment. We know that the cell walls have charged groups in it that can attract particularly cations. So cell walls tend to attract cations from their environment. So lots of interesting things that the cell walls do. Okay. One of the things that's present in the cell walls are the structural proteins. And the structural proteins, the largest groups of structural proteins are these HRGPs. HRGP stands for hydroxyproline rich. Glycoproteins. Hydroxyproline. Proline is an amino acid. Hydroxyproline is a common variant of proline, particularly in plants. And the hydroxyprolines here, the hydroxyl group, forms a place where glycosylation, attachment of sugars, tends to happen in these types of proteins. So the hydroxyproline-rich part of this means that these proteins are rich in places where carbohydrates can be attached. That attachment of carbohydrates to proteins makes it a glycoprotein. Okay? So there's a protein backbone in this, but typically there's lots of carbohydrate polymers that are attached to this. And it's the attachment of those carbohydrate polymers that gives rise to the covalent interactions that are important in cell walls. There's not a lot of them, but they certainly play a role, and we'll see what some of these roles are in just a minute. Okay, last thing I want to mention in terms of cell wall composition is when we think about secondary cell walls, one of the main components of the secondary cell wall that we talked about was lignin. Remember, lignin is a phenylpropanoid. It's a secondary product. But remember, the thing that we said that was interesting about lignin is it's these phenylpropanoids that are tied together through free radical reactions. That is, the formation of this network of phenylpropanoids is formed non-enzymatically, which means it's both strong and difficult to break down. The interactions of the lignins are not just among themselves. They can also interact with hydroxyl groups that are present in the celluloses and hemicelluloses of the cell wall. So basically, when you get to lignified cell walls, all the properties that allow the primary cell wall to stretch, to move relative to each other, is lost in the secondary cell wall because the matrix materials are covalently linked together. Right? So secondary cell walls prevent further growth of the cells, but also provide mechanical, structural strength to the cell walls that can withstand the positive and negative pressures, for example, that's present in the vascular tissues. Okay, I want to finish up by taking the properties that we've talked about for the primary cell wall and think about how those properties get back to this role that the plant cell walls play in dictating how cells grow, how cells enlarge, how cells change their shape, right? So everything we've talked about so far is a set of interactions that makes a cell wall that basically is pretty strong. You can generate turgor pressure within the cell, and it stretches a little bit, but it tends to not stretch very much, right? That builds up the turgor pressure that's present inside the cell. We need to think about how these interactions can be altered in order to allow the cell to grow. Because if we start off with this little meristematic cell with just a very thin primary cell wall, and that cell is going to grow to be a large epidermal cell or an even larger xylem or phloem cell, how does these basic properties permit these cells to enlarge and dictate how they enlarge, how fast, what their shape is, those sorts of things? Okay, to do that, we need to start off by thinking about how the role of the orientation of the cellulose microfibrils dictates the way a cell expands. Okay? And let's think of expansion just in terms of pressure dependent. So we increase the turgor pressure. We know that the cell is not a perfect osmometer. The volume of the cell increases. Some water moves in, right? That means the cell wall's got to stretch. So let's just first of all think about if a cell is stretching under increased turgor pressure, how does the orientation of the cellulose microfibrils dictate the way it stretches? So in the first case, we're going to start off with a cubic cell where all the cellulose microfibrils are randomly oriented. So let's go back again. So I should have put these next to each other, to this picture. And think about which way these microfibrils can move relative to each other. Can they move much this way? No, because the cellulose microfibrils don't stretch. Can they move this way? Yeah, because the things that are holding them together are not quite as strong. The interactions that hold these together are due to non-covalent hydrogen bonding, rather than this way, which is all covalent interactions. Okay, so what that means is, in cells where the cellulose microfibrils are randomly oriented, the cell tends to expand isotropically, uniformly in all directions. Where in a cell where all the cellulose microfibrils are laid down in the same direction, the cell tends to expand in directions that are perpendicular to the orientation of the cellulose microfibrils. Because you just told me the cell should be able to expand more that way because the interactions in the cell wall aren't as strong in the direction that's perpendicular to the direction of the microfibrils. Okay? So in the simplest sense, the expansion of a cell due to increased internal pressure we would expect to be in a direction that is perpendicular to the cellulose microfibrils. Can this picture that we've just described account for cell growth? What would happen to this cell if the only thing that's happened here is we've increased the pressure and the cell has stretched the wall under that pressure? What happens if we lower the pressure? Does it go back to this shape? Yes, it does. If it's only been the wall stretching. It's like a balloon. If you blow up a balloon, it stretches. If you let the pressure out, it goes back to its original shape. So what that tells us, if the cell is actually growing, something different must be happening. It's not just a pressure-dependent expansion, because that process is reversible. If the cell is growing, that growth represents an irreversible process. That irreversible process means some of the interactions that hold this wall together must be breaking, must be loosening in order to let the cell irreversibly expand. Right? So we can imagine, so here's this Hemi Cellulose that's holding these two cellulose microfibrils together. If we could weaken the interaction between the Hemi Cellulose and the cellulose, we could pull those fibers past each other, right? And then when we let them reform again, now we've got a cell that's larger. That's an irreversible change in shape. So it's really important that you make the distinction between reversible changes that are due to increase and decrease in turgor pressure, but no change in wall properties, versus irreversible changes that are associated with growth. Those irreversible changes represent rearrangement of the interactions among the properties or the components of the cell wall. If we think about this little meristematic cell that grows to be a big xylem cell, is the only thing that's present in that big xylem cell the wall that was present in that little meristematic cell? Okay, so the other thing that must be going on here is not just rearrangement of what's there, but adding in new wall materials as well, right? Okay, so we need to think about growth in terms of two things. Alteration of the existing wall to allow things to pull apart so we can have irreversible expansion of the cell. And we need to think about the incorporation of newly synthesized wall materials, because as the cell gets bigger, if we don't make new wall materials, the wall gets thinner and thinner and thinner. That doesn't happen. When a cell is expanding, new wall materials are being added at a rate that keeps the cell wall relatively constant in its thickness. So the primary cell wall, as the cell expands, the components of the primary cell wall are being continuously added to keep the wall at a relatively constant. Okay, so when we think about the irreversible changes in cell volume, the picture that we see is the same, because even though we can loosen up the interactions among the cellulose microfibrils, it still expands in a direction that's perpendicular more than parallel to the microfibrils. That should be reasonably clear from the fact that the cellulose microfibrils themselves don't stretch easily, right? So whether we're talking about pressure-dependent reversible expansion or whether we're talking about irreversible expansion associated with the growth, the orientation of the cellulose microfibrils is the key thing. Here we have random orientation, isotropic growth. Non-random orientation, anisotropic growth, growth along a specific axis. There's another component of this that we need to think about. In both of these cases, whatever is giving rise to the cell expansion is happening across all of the cell. But there are examples where cell wall growth happens only at a tip. Think of a growing root hair. Think of a growing pollen tube. It's not growth along the whole length, it's only growth at the tip. So, you know, the simple experiment where you put a bunch of dots along the tip of the cell or along a cell like this and that are evenly spaced. If it is diffuse growth, if it's growth that's happening over the whole surface of the cell, all of these dots should move apart by the same amount. If it's tip growth, only the ones at the tip move apart, right? So here we have growth that is localized. So we need to add one more thing to our list to think about. The loosening of the walls, the addition of new wall materials, and whether or not this happens over the whole cell or whether it happens in a particular place. Obviously, the signaling that's going on in this must be very different. Tip growth and diffuse growth have to have very different signaling going on. Okay, so we need to be thinking about that. All right. So, most of the questions that you guys asked in your reading assignment had to do with this figure. This figure is really important, and you understand it much better than you think you do. The questions that you asked were good ones, but they're questions that you know the answer to. So let's first of all just go over quickly what we're looking at here. In the bottom, we have information related to water potential and turgor pressure, right? So for a given osmotic characteristic of the cytoplasm, the water potential and the turgor pressure inside the cell are related to each other, right? You just add the osmotic potential to the turgor pressure to get the water potential, right? Okay, so these represent the characteristics inside the cell with regard to pressure. On the two y-axes, we have rate of water uptake or rate of cell expansion. And remember that the key thing in this figure was the intersection between these two lines, which we'll talk about in just a minute. The intersection between those two lines represents a balance between water uptake and increase in cell volume. The most common question I got from you, from the readings, is why is that balance point so important? So let's imagine a cell that is growing. It's increasing in volume. What would happen if the cell got big, increased in size, faster than the water came in? What would happen? Think about it. What's going to happen if the cell gets bigger faster than water comes in? Well, plasmolysis, what would be the characteristic of our cell under these circumstances? Not really plasmolyzed. Here's our original cell. The cell increases in volume this much, but only takes up that much water. What's this space filled with? How could it be filled with water if it didn't take up enough water to fill that space? What's it filled with? What's it filled with? Nothing. Nothing. Is that possible? No. Now, let's take the opposite. Cell increases by this much, and it takes up that much water. Is that possible? The cell takes up more water than the volume of the increase in the cell? No, water's incompressible, remember? That can't happen, right? So why is that point the only point that can represent the rate of growth of the cell? Because it's the point at which the increase in the volume of the cell is matched to the increase in the water content of the cell. Water can't come in faster than the cell expands, and it can't come in slower, because the cytoplasm is mostly water, right? Does that make sense? Yeah, yeah. So there's lots of reasons that neither one of these can work, right? So let's step back and think about what these lines represent. This one is relatively straightforward because this is just diffusion. This is basically just Fick's law. The rate of water movement from the outside to the inside of the cell is dependent upon the driving force, difference in turgor pressure, and the permeability. This LP is basically the permeability of the plasma membrane to water moving through it, and A is just the area of the surface area of the plasma membrane. So this line just represents the rate of water uptake as a function of turgor pressure. This line represents the growth of the cell, and the growth of the cell is being determined by turgor pressure trying to push it out, and the slope of this line represents how much the wall relaxes, how much it loosens up, how much those non-covalent interactions break in order to allow the cell wall to expand irreversibly. So the slope of this line represents something to do with how much that wall is loosening up. If it doesn't loosen up very much, it'll have a very shallow slope. If it loosens up a lot, it'll have a very steep slope. Okay? So these are two different processes. The loosening up of the wall that allows the cell to expand, and the movement of water that fills that expanding volume. They have to be the same. The only thing that can happen, the only growth that can happen, is the point where these two lines intersect. Okay? Make sure you understand that. That is really important. Okay, one thing that's important that we didn't discuss on this figure is this idea of a yield threshold. When people have studied plant cell growth, what they have found is that there's a minimum turgor pressure that's required for the cell to expand. That below this turgor pressure, there's no expansion of the cell. And that minimum turgor pressure is called the yield threshold. So only when the turgor pressure inside the cell gets above that yield threshold does the cell start to expand. Okay, so let's take a scenario. And think about how a cell is going to grow. So here's our cell at time 0. And let's just say at time 0, the turgor pressure inside the cell is less than the yield threshold. And we're also going to say at time 0, m is equal to 0. This factor that represents how much the cell expands per driving force, per turgor pressure, is 0. Okay? So, is this cell going to grow? Is this cell going to grow? Can't grow if m is equal to 0, right? It could expand reversibly if you increase the turgor pressure. But if you take away the pressure, it would just go back to the same size again. Okay? So the first thing that has to happen, if you just increase the pressure, the cell will stretch, but it won't grow. So in order for the cell to grow, we have to make this m greater than 0. We have to loosen up the interactions of the wall. Okay, so let's think about now, if P is greater than, if the Turgor pressure is greater than the yield threshold and m is greater than 0, what happens? What happens to this cell under these circumstances? It grows, right? It doesn't stretch reversibly, it grows because those wall interactions loosen up. The wall expands, the cell expands. When that happens, when the cell expands in response to the loosening of the wall, what happens to the Turgor pressure inside the cell? It decreases, right? Right? Because you've made the volume that it's enclosed in larger. So the Turgor pressure decreases. If the Turgor pressure inside the cell decreases, what happens to water uptake by the cell? If you lower the Turgor pressure, what happens to water uptake? What happens to the water potential inside the cell if you lower the Turgor pressure? Remember, decrease and increase are not good descriptors. Turgor pressure goes down, so is the water potential becoming more negative or less negative? More negative. What happens to water movement into the cell? It increases, right? So what that means is by lowering the things that are holding the wall together, increasing this wall extensibility, not only allows the cell to expand irreversibly, but it also provides the driving force for uptake of water. You've lowered the water, made the water potential more negative inside the cell by lowering the pressure potential. So water moves in, and when water moves into the cell, what happens to the Turgor pressure? Goes back up again, right? Right? So what has to happen to maintain this is a balance between the loosening of the wall, water moving in as the Turgor pressure goes down, the cell is expanding. These two things have to happen together, and it's controlled by the changes in M. We're not changing when cells are expanding. We're not changing the permeability of the plasma membrane. We're changing the extent to which the wall can irreversibly expand in response to Turgor pressure.
What determines M and how does it change?
Yeah, good question. So what determines M? So here's a simple experiment. We'll finish up quickly with this. Here's a simple experiment. We'll take a section of a plant from a seedling. We'll kill that section of the plant to basically remove all the biological components there. We'll put it in a little bath and we'll hang a weight on it, right? So that we're pulling on that section of basically dead cells, cell walls, right? And if you keep it at pH 7, it doesn't stretch very much. But if you lower it to pH 4.5, it stretches. No living cells here. This is just the cell wall. So the fact that acidification causes the cell walls to stretch under this pressure. If we go back to, here's the challenge for you for today. If you think about all the interactions, all the non-covalent interactions that hold this together, how does making the wall more acidic lead to a loosening of these interactions? I don't want you to answer it now. I want you to think about it from sort of a chemical perspective. Clearly it does. This experiment tells us that nothing more than acidification of dead cells allows the cell walls to stretch, to expand. Okay? And for a long time, plant physiologists thought that was it. Turn on the proton pumps, the cells expand. And they do, but they don't expand very much. Another similar experiment where you take these and you take your dead cell walls, but you take another piece and you basically extract stuff out of the cell walls without damaging it. In particular, they're looking for proteins that are in the cell wall. And if you add those proteins back to this guy at low pH, you see even more expansion and longer-term expansion of the wall than you do just by the addition of pH, the addition of acid. And by trying to run through all the proteins that are present here and separating them out and see which ones do that, they found that there's this group of proteins that are called expansins. Expansins are proteins that are released into the cell wall that are basically regulating the interactions here, decreasing some of these non-covalent interactions that allows the walls to expand. The details of how this works are just becoming available to us now. But we know that expansins are wall proteins, pH-dependent wall proteins that are necessary for growth of cells. If you mutate the genes for expansins, you don't get a normal plant because those meristematic cells don't get much bigger. The cells can't grow without the expansins. Okay, so that's basically the answer to your question. So one of the things that we need to do, one of the things I would, not for this exam, but for the final exam, is you should be able to tell me what's the difference between a cell that's growing isotropically and a cell that's growing at the tip in terms of all the things that we've talked about, right? Because all of these things, where the wall is loosening, where new materials are being added, and whether it is general over the whole cell surface or specific to a certain point, all of those things are going to control how the cell's going to expand. And now you have the tools to be able to understand that.